Hello everyone. So this is going to be a quick tutorial to talk with Nicole Warwick illustrations on the concept of the preganglionic and the postganglionic neurons. So there are a couple of ideas that we want to be familiar with as we go forward. Please ignore the obnoxious note about the window. So do recall that when we're naming neurons, it is important for us to consider the different names that are used for different systems. So we've previously discussed the somatic sensory system using first, second, and third order neurons. We also investigated the somatomotor neurons that include the upper motor neuron and the lower motor neuron, such as those we saw with the corticospinal pathways. Since we're investigating the autonomic nervous system, it's important to note that there are also a naming system in place for the neurons specific to the autonomic nervous system. Now, this works a little bit differently because the autonomic nervous system has various regions in the brain that are going to coordinate the autonomic nervous system. And those are going to include areas such as the hypothalamus with the cerebral cortex, areas of the brainstem, including pons, the medulla oblongata, and midbrain, as well as the spinal cord. So for our intents and purposes, we're not going to really look at the signaling pathways that come down through the brain. We're going to pick this up in the lateral horn. And so in the spinal cord, out to the target organ, there are, again, two neurons in sequence, much like we saw with the somatic motor system. Those neurons in sequence are going to include a neuron from the spinal cord to a ganglia, important piece of anatomy in the autonomic nervous system, and from the ganglia to the effector tissue. Therefore, the name is preganglionic for the neuron between the spinal cord and the ganglia, and postganglionic neuron from the ganglia to the effector tissue. We also have considered that the sympathetic and the parasympathetic systems are going to exit the central nervous system at different locations. So our sympathetic system is going to exit in the thoracolumbar area. That's going to be the purple and the reddish area tinged here on the spinal cord. And pathways from the sympathetic system are going to move into the sympathetic chain ganglia. So SCG here on the left side of the screen references that sympathetic chain ganglia. Outside of the sympathetic chain ganglia, on the parasympathetic side of things, we call this the craniosacral region because this autonomic nervous system division exits the cranial nerves, which are the peripheral nervous system features of the brain, and from sacral nerves, the very tippy top bottom of the spinal cord. These are also going to move out to organs and synapse in a terminal ganglia before going to the target organ. As another review, we did look into the sympathetic pathways. 3 are concretely recognized by all instructors and textbooks. 1 is presented differently depending upon who you're talking to. For the sympathetic pathways, you have all of your preganglionic neurons illustrated in the brownish colors, starting here in the lateral horn, moving through the ventral horn, out ventral rootlets, and the ventral root to the spinal nerve. From the spinal nerve, all of these pathways are going to enter into the white communicating rami and enter into the sympathetic chain ganglia. Some, like the spinal nerve pathway, will synapse where the information entered and then quickly move through an unmyelinated neuron through the gray commissure or gray communicating rami and out to effector tissues. These tend to go to the somatic motor and skin to help control things like erector pili muscles, controlling your hair standing on end, sweat glands, blood vessels in the skin, and blood vessels in the muscles, the skeletal muscles. Another pathway known as the sympathetic nerve is still going to have their preganglion entering through the spinal nerve through the white communicating rami, and here it's going to move up. It's going to go superior in the sympathetic chain ganglia to other regions where it will synapse and send a signal out a neuron unmyelinated postganglionic neuron, and these largely go to things like the salivary glands, to control the diameter of your pupil, to control the smooth muscle tone in your airways, your heart, the esophagus, and blood vessels in general. Then another route known as the Splenic nerve route, still starting in the lateral horn, ventral rootlets, ventral root, spinal nerve out and wrapping through the white communicating rami. This particular neuron is going to travel through, but not stop or synapse in the sympathetic chain ganglia all the way out to a collateral ganglia, where the postganglionic neuron synapse is located. And there we're going to target things like the liver, the spleen, we'll see the stomach, you can have the adrenal gland, kidneys and bladder, as well as reproductive organs. And I've included a fourth pathway here just as a review for the sympathetic system, that is the adrenal route. And here, a preganglionic neuron comes out in the same fashion that the other pathways have. It sweeps through the white communicating rami, bypasses the sympathetic nerve system, just traveling through, travels through the collateral ganglia as well, and terminates in the adrenal medulla, or in the adrenal gland, where it's going to cause the release of both epinephrine and norepinephrine to a lesser degree, and even a lesser degree, tiny bits of dopamine. These will be released into the bloodstream and will act as a hormone signal to other areas of the body. So the goal of what we want to talk about is really to investigate the synapses and to look at the neurotransmitters and the receptors that we find both in the sympathetic system as well as in the parasympathetic system. So we're going to start with this particular illustration, and I've drawn in some of the neurons, but we have some more details that we can add to this. So what we want to do is look at the synapses, and because the autonomic nervous system is using a preganglionic and a postganglionic neuron, what that means is that there are two synapses that become important to us. We want to investigate the synapse inside the ganglia. That is going to be the synapse between the parasympathetic preganglionic neuron and the postganglionic neuron. We're going to do this for the sympathetic system too. So one of the things that may be important to note, something to keep our eye on, is that we're talking about the terminal arborizations of the preganglionic neuron, and we're talking about the receptors in the dendrites that attach to the soma of the postganglionic neuron. And so just to review from long ago, the concept that we have neurotransmitters that sit inside the terminal arborizations that are going to release neurotransmitter into the gap, into the cleft, the synaptic cleft between the preganglionic neuron and the postganglionic neuron. Here, we're going to have channels, receptors, ligand or ligand gated channels that are going to respond to the neurotransmitter. So the neurotransmitter will engage the receptor, and this should depolarize the cell. It should create a local potential sufficient to hit threshold so that the postganglionic neuron sends a signal. Now, what's important to note here is that this particular synapse between the preganglionic neuron and the postganglionic neuron, do we need to write that in? Why not? Wouldn't hurt, right? Let's use a color virtually unheard of, hot pink. Here's my preganglionic, and here is my postganglionic. This dotted line around this synapse is supposed to represent things that are occurring in a ganglia. And you may recall or not that the specific ganglia we're talking about in the parasympathetic system is called the terminal ganglia. So here I have my first synapse, neurotransmitters released, and you may notice in the black right here that we have a nicotinic label. This is the nicotinic receptor that you have been introduced to. Nicotinic receptor. This responds to acetylcholine. So we get acetylcholine being released to bind to that nicotinic receptor. Now, of course, there are other compounds like nicotine that could also bind to this receptor. That's going to create the action potential here in the parasympathetic system. Postganglionic neuron is carrying the action potential from the terminal ganglia out to the effector cell. And that effector cell, as part of the parasympathetic system, could be in the heart, it could be in the gut. There's a variety of tissues connected to the parasympathetic division. What this is going to do is then cause an action potential to arrive at the terminal arborizations right here in this second synapse that we're going to consider between the postganglionic neuron and the effector tissue. Again, neurotransmitter in here, in this case, also being acetylcholine. So maybe I ought to bring in my acetylcholine, but I'm going to do it in green just to highlight the fact that the receptor here is muscarinic. So this is really using the same neurotransmitter. Same NT, okay? And so what happens is, to quote previous courses, an action potential arrives at the presynaptic, because this is the synapse, the presynaptic terminal of the postganglionic neuron. Voltage-gated calcium channels open, which allows calcium into the cell, causing exocytosis of acetylcholine into the terminal cleft. The acetylcholine in the cleft diffuses through the cleft to the muscarinic receptors located on the effector organ. Acetylcholine binds to the ligand-gated channels and stimulates response in the effector. So the way the parasympathetic nervous system works, which remember has a very narrow effect, is the preganglionic neuron that exits the central nervous system and heads to a ganglia, in the case of the parasympathetic system known as the terminal ganglia, then releases acetylcholine that binds to the nicotinic receptors. This creates a local potential. When it hits threshold, triggers an action potential down the postganglionic neuron, sending the signal out to the effector organ. In the synapse that occurs between the postganglionic neuron and the effector, more acetylcholine is released to bind to the muscarinic receptors that are found on the effector organ. So this summarizes the methodology behind the synapse sequence in a parasympathetic neuron, the two synapses in the parasympathetic system.